This presentation is going to go through the foundational concepts that are tested in the biological and biochemical foundations of living systems sections on the MCAT. The disciplines that are tested are going to be first semester biochemistry, intro biology, gen chem, and organic chem. The percentages are approximate within 5%. So, for example, a test may be 30% biochemistry or 20% biochemistry. So, an average of 25%, depending on the types of questions that are in the section. But you will recognize more than anything that the intro biology is 65% of this exam, of this section on the exam. When they say intro biology, they're not just talking about the classes that you take in the first and second semester of your biology courses within college. These are also talking about anatomy and physiology, microbiology, and some other concepts that may be briefly covered, but not in the amount of depth that the MCAT is testing. So taking some of those advanced courses is also helpful for getting through the content on the MCAT. It is not required, but it is helpful when trying to help understand all of the concepts in the depth that MCAT is testing them. Foundational concept number one is going to be about 55% of what is covered on the MCAT. And it says, biomolecules have unique properties, determine how they contribute to the structure function of cells, how they participate in processes necessary to maintain life. That's a long sentence. And a lot of those words are created in like a parallel structure situation where things go together, but you have to be able to break up and understand what they mean. When I'm going through this statement, it helps for me to be able to define each term individually. So biomolecules, when I think of biomolecules, I think of the 4 macromolecules that are covered predominantly in biochemistry, but also in some other courses. So I'm thinking about those 4 macromolecules. I'm also thinking about the structure of cells. So it says biomolecules, unique properties determine how they contribute to structure and function. Okay, so structure and function makes me think that there are 2 things it's talking about: the structure of cells as well as the function of cells. So I need to understand both the structure of a cell as well as the function of a cell. And then also how they participate in processes necessary to maintain life. So for me, I think about what are the processes that are necessary to maintain life. And so when going through and even developing what I'm going to review when studying for the MCAT, understanding each of these terms in depth will really help me figure out what gaps I have and what things I do feel already strong in that I don't need to go back and cover in as much detail. So let's talk about biomolecules. The 4 macromolecules, there's amino acids and proteins, nucleic acids, lipids, and then carbohydrates. I think about these 4 macromolecules when I'm thinking about all of the rest of the things that are stated within this foundational concept. So then when it talks about the cell structure, what kind of cells are we talking about? We're talking about eukaryotic cells, but we're also talking about bacterial cells as well as some types of viruses. And I have to know the structure of all of those things. Okay, then I'm also going to have to understand differences. So how are the macromolecules talked about previously? How are each of those macromolecules going to be structured in each of these cells? That's the beginning of the integration that's going to be tested on the MCAT. So is bacterial DNA structured the same way that eukaryotic DNA is structured? Do lipids exist in a viral capsid? If they do, what types of lipids are there? Are those the same lipids that you would see in a eukaryotic cell? These are some of the types of questions that I would encourage you ask yourself when challenging yourself to integrate these concepts for the MCAT study. When it comes to cell function, what kind of cells? Again, I'm talking about bacteria, eukaryotic cells, as well as viruses. And we're talking about their functions, not their structure. So now, how do all of those things work? And again, the macromolecules talked about before, how are those macromolecules fitting into each of these different things? And what macromolecules are going to be different to help each individual cell function? For some examples of questions, what in RNA does a virus versus eukaryotic cell. What's it going to do? I know in a eukaryotic cell, RNA helps with mRNA, it helps with making proteins happen. But within a virus, it might do other things. Asking yourselves those types of questions. How does bacteria use glucose? Okay, I know for the most part, I understand glycolysis and I understand the citric acid cycle and I understand the electron transport chain. But does bacteria go through all of those steps when it's using glucose? Does it do other things? These are just some of the questions you can be asking yourself when challenging yourself to integrate the concepts. Then we have processes to maintain life. What are the processes that are necessary to maintain life? Things that I can think of. You need to breathe, you need to eat, you need to sleep, your heart needs to beat, and you got to move in some way to be able to access food, for example, to get away from danger. So some type of movement has to occur. And again, how are the macromolecules contributing to each of these processes? So some questions you could ask yourself. Muscles, I know, are made up of mostly proteins. Muscles contract to help you breathe. So then I can think of proteins in terms of processes to maintain life. I also know some lung cells have membranes to transport oxygen and carbon dioxide. So they're going to have specialized membranes. Does that mean they're going to have different lipids or a different structure in its cell membrane compared to other cells? Those are all eukaryotic cells, but what's going to be different about some lung cells? And I know some other lung cells are going to produce mucus to help keep infections out. So if I'm thinking about infections, I'm also thinking about immune cells. And so what things are going to have to be integrated for all of those processes to occur? So going back to the foundational concept, this is what it's stated in AAMC words. And so if I'm using my own definitions, how would I state this foundational concept for myself? Each of the 4 macromolecules has unique properties. Those properties contribute to how bacteria, viruses, and eukaryotic cells look and work. And then those properties also help keep cells alive. This helps me understand it a lot better than what the AAMC said. And when you're going through the MCAT and answering questions, you're going to see questions structured in a way that may not be comprehensible for you, and that if you put it into your own words, it would make a lot more sense. So I really encourage you, both when studying these concepts as well as when practicing questions studying for the MCAT, write the things in your own words as much as you can so you can keep track of what is actually being said and you don't have to go back and redefine those things every single time you read that question. You feel more comfortable with your own words than with somebody else's words. When you're studying, when you're going through, the MCAT's going to be asking you a lot of questions based on how you understand the concept, based on your understanding. It's all multiple choice. It's eliminating things that are not correct based on what you know to be correct and what you know to be exceptions that might be true, might not be true, just eliminating things that are absolutely not true. And so in order to answer MCAT style questions, you're going to have a really strong understanding from the beginning to the end, from an atom to an organism, of how macromolecules work. And this is the beginning of integrating the concepts that you previously learned separately. So you may have learned some of these concepts within one biology course, some of these concepts in a different biology course, some of these in a chemistry course, but these are all going to be integrated when it comes to taking an MCAT test and the types of questions that they're going to be asking. Within the foundational concept, they also break it down. So category 1A, they state as construction, function, properties, constituent amino acids. Okay, so I anticipated that this was going to be about the 4 macromolecules, and here we go. Here's one of the macromolecules being discussed, the amino acids and proteins. Again, I'm going to break down this category into smaller concepts for me to be able to really define what I'm looking for in things that I know and do not know to help organize my studying. So the structure of amino acids, they're also the structure of proteins, and then also the function of amino acids and proteins. So first thing we need to define is amino acid structure. the general structure, which AAMC defines as an absolute configuration at the alpha position, that being defined by R group variations. Then also those R group variations lead to how amino acids are classified. So if you have polar amino acids, non-polar amino acids, hydrophobic, hydrophilic, other different special types of amino acids, are there any that have special structures, unique structures that contribute to different things within an enzyme being able to function? You're also going to know about protein structure. So, okay, how does primary structure occur? Via translation. Do I understand how translation works? Secondary structure, tertiary structure, and quaternary structure. I'm also going to have to know, okay, if that's how you build up a protein, how do you break down a protein? So denaturing of a protein, how does that occur? Also talking about isoelectric points. So if we go back to the amino acids, knowing how different R groups create different charges, there's some point where it's an isoelectric point. Do I understand how to calculate an isoelectric point? And do I understand what an isoelectric point means when it comes to how an overall protein is structured compared to an amino acid itself? Then also the function of amino acids. So where are they located within a cell membrane? If I have a hydrophobic amino acid, where's it going to be compared to hydrophilic amino acids? If we have phosphorylation of the amino acids, what amino acids are actually able to be phosphorylated? We talk about phosphorylation of proteins, but if we know proteins are made up of amino acids, those amino acids are actually things that are getting phosphorylated. So which amino acids can participate in phosphorylation as well as acetylation? Acetylation has to do with histone forming and access to genetic material. So understanding which amino acids can be acetylated, deacetylated in order for the actual protein to function or not function is also really important for the MCAT. Then how proteins function. So proteins as enzymes, most of the time we think about proteins, and when we think about enzymes, we know all enzymes are proteins, but not all proteins are enzymes. So the MCAT is also testing, do you know about proteins being enzymes? What enzymes do? Enzyme kinetics, which deals with the Michaelis-Menten equation, inhibitor types, so competitive, non-competitive, etc., as well as proteins not as enzymes. Do you understand what else proteins can do? So if you have proteins involved in cell structure, do they help with the receptors? Do they help with the rigidity of the cell membrane? Do they help with the cytoskeleton, other parts of the cell structure? They're involved with with muscle contraction for sure. So how are proteins involved with muscles? They're also used as receptors within a cell. So those are other things proteins can do. That's not an exhaustive list. None of this will be exhaustive, but those are some of the things that proteins do within cells as well. Then you're going to also have category 1B, where it says the transition of genetic material from gene to protein. Okay, I knew one of the macromolecules that we were talking about in this foundational concept 1 was nucleic acids. And so genetic information makes me think of nucleic acids, gene to protein. When I say gene to protein, that's for sure the central dogma. So central dogma is transcription and translation, but those things are going to be different within eukaryotes, bacteria, and viruses. If I'm thinking again back to the different types of cells, thinking back again to structure and function of amino acids and proteins, how those worked in different ways within a cell, thinking here as well. Within each cell, how are those things going to be functioning, but also within different types of cells, how is it going to function? You're also going to have processing. So it's not just transcription. You also have post-transcriptional modifications where you have introns and exons and splicing. You also have modifications of proteins as well. And so the protein packaging, if it's going to be shipped out or exported from the cell versus staying within the cell, how are those proteins going to be processed differently? If the protein's becoming an enzyme, how does that work? Then also talking about the nucleic acid structure. This is talking about, okay, DNA versus RNA, knowing all of those nucleic acid bases, but also which nucleic acids are going to be present in which types of cells. Are there any unique properties? For example, thymine versus uracil. Where do you find which and why? If you think about the structure of uracil, what other what other nucleic base is it most similar in structure to? Those are things that the MCAT will be testing not explicitly, but assuming you have an understanding of those things, they'll be asking questions to integrate those concepts. You have category 1C, transmission, heritable information, generation, processes, increasing genetic diversity. So previously we talked a little bit about nucleic acids and their structure and function, but this is also going more in depth about nucleic acids from generation to generation. So heritable information, talking about transmission of DNA from generation to generation. This is also genetic diversity, increasing genetic diversity. So if we're talking heritable information, we've got genetic material, DNA, RNA. Are there other types of genetic material for different types of cells? What else can occur? How are those things transmitted? How is RNA transmitted from generation to generation, DNA, etc.? If we're creating a new being, how does meiosis have to do with that? Depending on what type of being we're creating. Are we creating a new bacterial cell? Are we creating a new leaf? Are we creating a new human? There's going to be differences in how those things happen. There's also genetic diversity. So a genotype versus a phenotype. If you know back to the previous concept where it was talking about DNA structures, if you know introns and exons within post-transcriptional modifications, what is the difference between your genotype and your phenotype? If you were talking evolution over generations, how does natural selection, survival of the fittest, other concepts associated with evolution contribute based on DNA transmission? And this is really where meiosis comes into play when talking specifically about how humans create different types of humans. Crossing over is one concept that is really assumed you have an understanding of, because there are a lot of diseases that can happen within humans that are because things are not crossed over appropriately. So understanding how crossing over happens, what stage in meiosis it crosses over, and especially in relationship to the cell cycle, the differences that occur there. Then 1D, you're going to have principles of bioenergetics, fuel molecule metabolism. Bioenergetics is one word that I know I'm going to need to define. That I think of as transferring energy. I think of bioenergetics and enzyme kinetics often interchangeably. So this is still talking about how enzymes transfer energy and like that delta G, Michaelis mentioned some other functions of enzymes. And then it says fuel molecule. So when I think of a fuel molecule, most of the time I think of glucose, but I'm really thinking of any molecule that can make energy. So glucose I know of is one molecule, but are there others? And also the metabolism. So within eukaryotes, bacteria, viruses, is their metabolism going to be different? If they are metabolizing different molecules and make energy, how are those molecules going to be created? But also, how are they going to be broken down within different cell types? And then you're going to have foundational concept number 2. This one is about 20% within 5%, so 15 to 25% of the Biochem section of the MCAT. And it is defined as highly organized assemblies of molecules, cells, organs interact to carry out the function of living organisms. Now, that's, again, a long sentence. Even within me reading it, I was trying to restate it to make it make sense to me. And so this is where it really helps to just break it into smaller words and then define those smaller words for myself to then have an understanding of the concept overall. So assembly of molecules, what does that mean? So if I've got a single-celled organism, how are things going to be assembled versus multicellular organisms? Then we're going to have assembly of cells. So if I have assembly of cells, that's clearly not going to happen in single-celled organisms. We're going to only have that in multicelled organisms. And so how is that going to occur? Any multicellular organism is most commonly referred to as eukaryotes. And so really thinking about eukaryotes, how are cells assembled into different things within eukaryotes? And then you also have assembly of organs. So within humans specifically, you have a lot of different organ systems that are different assemblies of different cells, and those cells are all structured differently. So it's also specifically testing about the human body. And then each of those living organisms functions. So living organism function makes me think about the first concept and back to some of those concepts that were discussed previously. So this is, again, where things start to get integrated because, okay, I see concept 1 coming into concept 2, and concept 2 could be reflected some other places within concept 1 if I had seen these in a different order. So then 2A is talking about assemblies of molecules, cells, groups of cells within single cellular, multicellular organisms. Okay, so assembly of molecules in single-celled organisms, as well as assembly of cells within multicellular organisms. And those multicellular organisms are coming from molecules within single cells. And then you're also going to have assemblies of groups of cells within multicellular organisms. You're not going to have a group of cells in a single-celled organism. So again, that's just within multicellular organisms, and breaking down what each of those mean. So an assembly of molecules, we're going to talk about bacterial cell structure. Those are often single-celled organisms, almost explicitly single-celled organisms. We're going to have gram-positive as well as gram-negative bacterial cells. What's the difference between the two of those? Also, within bacterial cells, how does cell transport occur? So if a bacterial cell needs to move something within itself, how is that transport going to occur in comparison to how would it occur within a eukaryotic cell? We're also going to talk about viral cell structures. So viruses are not considered cells, so viral structure. But as it relates to breaking down this foundational concept, remembering viruses are also part of this. So they've got the capsid, they also got some type of genetic information, and viruses most of the time have to do some type of cell transport as well. So how would a virus do cell transport? Would the virus have to use its host in order to do transport within itself? Or would it be able to do other things in order to do transport within and of itself? Then we've also got eukaryotic cells. And how are they structured? What's different between eukaryotic cells, bacterial cells, and viruses? Their organelles, what types of organelles, where do they come from? What are the hypotheses of why those organelles exist? And how do those organelles function in eukaryotic cells now? As well as their plasma membrane compared to a bacterial cell wall and a viral capsid. What does a plasma membrane of a eukaryotic cell look like? And we're also talking about cell transport. So how does cell transport happen within a eukaryotic cell? Then assembly of cells, multicellular organisms. So just talking eukaryotes here. So we're going to be talking about cell junctions. We're also going to be talking about cell-to-cell communication. We're also going to be talking about cell transport between two different cells. So if we've got one cell set up to make something for another cell, how does that cell transport it to another cell? And then we've got assemblies of groups of cells. And so once we've got a bunch of cells that are grouped together into something called a tissue, how do those tissues communicate together, make up an organ system? Each of those specialized tissues is going to do something else within an organ system. And so how do those organ systems also work together? We have cell signaling. Is this going to be a short cell signal, a long cell signal? What's the difference between those two things? Then we're going to have category 2B, the structure, growth, physiology, genetics, prokaryotes, and viruses. We're going to have structure of prokaryotes. We're going to have the growth of prokaryotes. We're going to have the physiology of prokaryotes. We're going to have the genetics of prokaryotes, structure of viruses, growth viruses, physiology of viruses, and genetics of viruses. Structure of prokaryotes and viruses, we've already covered within 2A. So this is something where, again, you see how this is starting to get integrated in other concepts. But if you don't know the structure, you're not going to understand physiology and genetics and other things within viruses and prokaryotes. When talking about prokaryotes, they're speaking about bacteria as well as archaea. So growth and physiology of prokaryotes. If we're talking growth, that has a lot to do with the metabolism. Prokaryotes are all single-celled organisms. So their growth is going to be different than how eukaryotic organisms would have growth. It's also going to be talking about physiology. So how does cellular respiration occur within prokaryotic organisms? How is it different? How is it similar to eukaryotic organisms? As well as reproduction. if we have cells that are going to reproduce, but they're single-celled organisms, how is that reproduction different than what it would be within a eukaryotic cell or a virus? As well as symbiosis. So can these cells live on their own, or do they need something else in order to be able to live? If they need something else in order to be able to live, what type of symbiotic relationship is it creating? Do I understand the different relationships? We're also going to have genetics of prokaryotes. So this is where we're talking about plasmids, but also extra genetic material. So there's going to be the bacterial DNA and other things besides the DNA itself, or RNA. Then also conjugation. So the prokaryotes, bacteria especially, are not going to just stay as the same genetic material they started with. A lot of times they will be able to conjugate and share their genetic material between the two of them. How does conjugation occur? There's also transformation. That's another way they can change their genetic material. How does transformation occur? Transduction is another way that they can change genetic material, not necessarily with another bacteria, with something called a bacteriophage, which is just another name for a virus. But how does transduction occur? Do you understand this in the genetics of how prokaryotes, both archaea and bacteria, work? Then you have growth and physiology of viruses. So we're going to have the life cycle of virus. We know viruses are not cells. They cannot live on their own. They need something else in order to be able to live. So how does the viral life cycle occur? Not a lot of viruses have the same life cycle. What's different about the different life cycles viruses may have? Bacteriophages is, again, we were talking about transduction previously, and that has to do with viruses being able to change bacterial DNA, and that has a little bit to do with like the whole host situation. But how does viral transduction occur? Then there's also retroviruses. So some viruses are going to be retroviruses. They're not going to just use a host to do normal creating its own RNA. They're going to create some DNA. So do you understand how a retroviral DNA creation occurs and why it's needed within a virus? And then there's also going to be asking about things that come from viruses, or not viruses in and of themselves, but are like virus pieces. So we've got prions as well as viroids. Do you know what those are? Do you know how those work within cells, within eukaryotic cells and bacterial cells? What's the difference? How do those impact the way those cells are able to function? We're also going to have the genetics of viruses. So is it RNA or DNA? And does it have an envelope or not? That's a lot of the way that viruses are categorized when thinking about how they're able to infect eukaryotic and bacterial cells. Then you're going to have concept 2C, where you have processes of cell differentiation, division, and specialization. So again, breaking this down, you've got cell division, making sure you understand how that works, cell differentiation, and cell specialization. So what is cell division? The eukaryotic cell cycle, recognizing that that's going to be different from the viral life cycle, as well as how bacteria are able to replicate and make new bacterial cells. The control of the cell cycle is really important, because the control of the cell cycle has a lot to do with mutations if mutations occur, that control is not occurring, and that control happens predominantly through checkpoints. So do you understand what those checkpoints are within the eukaryotic cell cycle? And if those checkpoints aren't hit, what may happen to the cell itself, and what may that then lead to within a multicellular organism like a human? So if we're talking about P53, for example, with one of those mutations occurring, how that would lead to cancer. Do you know the difference between an oncogene, proto-oncogene, tumor suppressor genes, other things that have to do with the genetics within the eukaryotic cell cycle, and how specifically it leads to cancer? Do you also know about cell senescence? So if a cell is not in active replication, it's not going to create another one of itself. It's just in the G0 phase. Do you understand what types of cells are more likely to be in the G0 phase? Why those cells are likely to be in the G0 phase? What would happen if those cells leave the G0 phase? What triggers something to leave the G0 phase? Do you understand how all of those things work? That all has to do with some types of diseases developing within the human body. We've also got the mitotic structures. So you know what things make up the structures associated with mitosis. It's not just the DNA that works within mitosis. You've also got the spindle fibers. You've got the centrioles. Do you know how asters are created? All of the other things associated with mitosis as well. And then how gametogenesis, or the meiosis, where you're going to be making gametes, sperm and egg cells, how that's going to be different in a cell cycle when you're making sperm and egg cells. So what cell cycle differences are you going to have when making sperm and egg cells, or gametogenesis? How is gametogenesis going to be varied in the cell cycle? Then you've got cell differentiation. So if you start with pluripotency, you know, once you've got the sperm and egg joining together, it just makes a zygote and eventually makes a fetus that becomes a baby, and those cells have to differentiate over time. But it starts with pluripotent cells that can become more than one different thing. So starting with stem cells, do you understand what cells are more likely to be some stem cells? Can you induce stem cells from types of cells? What types of cells can make more pluripotent cells if stem cells are induced? And what cells cannot be made into some type of stem cell because it's too far into differentiation? Then also the signals for differentiation. So if you have a blood cell that needs to be made, a red blood cell specifically, it's going to start from some type of stem cell that is pluripotent, and then there's different signals that tell that cell it needs to become a red blood cell. If those signals don't occur and different signals occur, it might become a white blood cell, it might become an immune cell. And so recognizing what signals for differentiation help stem cells or pluripotent cells become into differentiated cells with one specific type of function. This is really important when it comes to embryogenesis. Then you've got cell specialization, where you know if you have a multicellular organism that's made up of different tissues, all of those cells are going to have different functions. Do you know how that's going to happen based on what tissue type you have, what organ system you're starting with? All of those things are going to be covered in foundational concept number 2. Then foundational concept number 3 is the rest of the Biochem section. So about 20 to 30% of the Biochem section of the MCAT. And it is, again, a long one. The complex systems of tissues and organs sense the internal and external environments of multicellular organisms, and through integrated functioning, maintain a stable internal environment. Breaking that up, I see systems of tissues and organs. I see internal and external environments of multicellular organisms, which is still a long sentence, but I can break that up later. And then I also see systems of tissues and organs have integrated functioning. And then those systems of tissues and organs also maintain a stable internal environment. So those things are still pretty large concepts, but can be more easily broken down than that large sentence by itself. So structure and functions of the nervous system and endocrine systems, and ways these systems coordinate the organ systems. A really long statement. So again, structure of the nervous system. We've got structure of the endocrine system, and we've also got function of the nervous system and function of the endocrine system. And then also, how do those coordinate other organ systems? So structure of the nervous system, we've got the brain and the spinal cord, the axon structure, supporting cells that go with axons, whether we're in the PNS or the CNS. Do you know which cells are in which? Sympathetic versus parasympathetic divisions, other divisions of the nervous system. Some of those divisions have to do more with the function than the structure, but really knowing CNS versus PNS, the brain versus the spinal cord, and then the sympathetic versus parasympathetic. CNS and PNS are also known as the central nervous system and the peripheral nervous system. And then you've also got sensory and effector neurons. So within the spinal cord and the neurons that go out to the rest of your body, some of them are just sensing and some of them are making things happen. Do you know the difference between which ones? And there's also reflexes if you've got those systems happening. Do you understand how that happens? Then you've got the structure of the endocrine system. So you've got the primary and accessory organs of the endocrine system, their anatomy, as well as the hormones that each of those organs are making. What do those hormones do? And when we're talking about biosignaling, how do those hormones lead to telling another cell to do something else? So is that going to be like a G protein coupled receptor, or some of the other types of receptors that have to do with biosignaling? The function of the nervous system has to do with signal transmission as well as nerve conduction. So do you know more than anything how concentration cells lead, how the concept of concentration cells contributes to how nerves actually function? If we're talking about internal and external environments, what is on the inside of a cell, what's on the outside of a cell, how does that lead to an action potential? Where is that potential occurring? If we need a transmission to occur, how does an action potential lead to signal transmission? All of those things are part of the function of the nervous system. But we're also going to be talking a little bit about reflexes. And so if we're talking about the effector and sensory neurons, that goes back into the central and peripheral nervous system when we're going to have reflexes occurring. And you can see with the concentration cells, that's also where a little bit of the general chemistry or some of the chemistry concepts are going to be tested within the Biochem section of the MCAT. It's not just one thing. All of these things do become integrated into making the human body work. So really understanding how the integrated functions happen is necessary to be successful in the MCAT. Then we've got the function of the endocrine system. So you've got signal transmission. You've also got the second messengers. So if we've got a GPCR, what is the second messenger system? How are the second messengers going to occur? What are the steps within the second messenger system? What other things those lead to happening? If we're going back to proteins, what proteins are there? Are those enzymes? What reactions are occurring? Recognizing all of those things within the function of an endocrine system if we're talking about hormone transport. And as well as homeostasis. So we recognize this is all having to do with like sensing the environment, the internal and external environment. And so the internal environment is part of that maintaining homeostasis. And homeostasis is maintaining what you've got going on, like making it not change itself. But do you know how the endocrine system helps your body maintain homeostasis of every different organ system that is in the human body? And that leads to the coordination of the other systems. So we're talking biosignaling. If the endocrine system's trying to tell the digestive system to do something, how would that biosignaling be different than if the endocrine system's trying to tell the musculoskeletal system to do something? There are all of those hormones. So if the endocrine system's sending hormones, are there other organs that are needed in order for those hormones to be created or secreted? Are those other organs where those hormones are stored? How do those hormones work with other organs? As well as the other organ systems also help maintain homeostasis. So how does the endocrine and nervous system help the whole entire body made up of different organ systems maintain homeostasis? And then you've got 3B, where you've got structure, integrated functions of the main organ systems, where breaking that up, you've got the structure of main organ systems, as well as integrative functions of main organ systems. When I think of the structure of main organ systems, the main organ systems that I'm thinking about are the respiratory system, circulatory system, the lymphatic system, the immune system, digestive, excretory, reproductive, skeletal, and muscles. Each of those, I understand the structure of their system. So if I'm talking about the circulatory system, we're talking about the heart, but not just the heart and the valves of the heart and the 4 chambers of the heart, but also all of the arteries, all of the veins. Do I know how blood flows within the human body outside of the heart as well as within the heart? We're talking the digestive system. Do I understand from the mouth to the anus, how does the digestive system work? just in its structure. And then we're talking integrated functions. So yes, each of those organ systems works in separation, but a lot of how the human body works is it's not just working because it's working itself. It worked because something else told it to work. It's working because it's helping something else work. So how are all of those functions integrated within another? So we've got the respiratory system is going to work with the circulatory system. If I need blood to flow, I'm going to need to breathe more, because the reason blood is flowing is for oxygen to get delivered to cells. So those work together. If I have the immune system, okay, I can make immune cells, but the lymphatic system has to be there in order for that immune cells to transport. The lymphatic system also has to then work with the circulatory system, and seeing if there's other integrated function. We know the musculoskeletal system is often referred to instead of just skeletal system or muscle system, because they work so much in integration. So challenge yourself. Are there any other integrated functions that you can think of, and how would those integrated functions work in the human body to help it maintain homeostasis? So then the MCAT also talks about something called SIRS, which are the scientific inquiries and reasoning skills. There's 4 skills that the MCAT assumes or tests students on, and those skills will be covered in depth in another video. But all of those SIRS are how the questions can be presented. So if it's talking about a math concept, it can be presented as asking that. If it's asking about data analysis, it can be asking a question in that presentation. The SIRS help you understand how this question was presented, but the foundational concepts are the things that it was testing. So for example, if it was testing your understanding of the integration of the respiratory and circulatory system, and it was testing that through a chart or a data analysis, it could give you data that shows you different oxygen levels and how much the heart is beating with those different oxygen levels. And then it would ask you a question about when is the heart beating the fastest, and you have to base that on a different oxygen level. That's just one way the MCAT could ask a question based on the foundational concepts being crossed with a SIRS. Again, the SIRS will be covered in depth in another video. Thanks for watching.